So second, by prevalence, parenteral hepatitis is hepatitis C. Again, similar to oral hepatitis, hepatitis C resembles hepatitis B, and by clinical presentations, very close to hepatitis B. The major difference in B and C hepatitis is virus causing hepatitis C infection is single-stranded RNA-containing virus. It belongs to family of Flaviviridae, and nowadays it is classified as Hep C virus, and hepatitis C virus is the name of the species. So the classification changed for several times in the last decade. So maybe in some of the books or in some papers you will find it as belong to another family or named by different way. But that is the last classification till the moment. And in the contrast with hepatitis B, that virus contains RNA genome, and complete eradication of disease from human body is possible. So nowadays, more than 70 subtypes of viruses are described, and similar to hepatitis B infection, two types are possible: acute and chronic hepatitis. And in the contrast with hepatitis B, that virus is much less contagious and usually requires direct contact with the blood. So very rare cases of sexually transmitted infections and some other contacts are described, but usually it is direct injection of blood or sharing needles in drug abuse or some other tissue donation is the major way of transmission of that disease. So again, prevalence is provided here. It is more actual data, and again, mortality rate is about 400,000 people annually because of the same salutary complications, liver cirrhosis and hepatocellular carcinoma. So hepatitis C is prevalent in Eastern Mediterranean region and in European countries. So we have nowadays several drugs for therapy of hepatitis C infection, but they are quite expensive. So if you talk about structure of virus, virus is enveloped in structure of virus. Two major proteins are identified. It is enveloped protein 1 and enveloped protein 2, or E1 E2. And capsid is made of additional protein, which is named as a C, and RNA genome is packed in the capsid. So both enveloped proteins E1 and E2 are essential for infection and fusion with the host cell. As well, F protein is described of frame shift protein. It is involved in persistence of virus in host tissues. And usually antibodies against that F protein are identified in people who have chronic infection with hepatitis C virus. So I will not talk about clinical presentations of the disease because they are identical to classic viral hepatitis. We have screening tests for antibodies against core antigen, and if you need to confirm, it is usually used as a screening test. And if you need to confirm the disease, then you have RNA of the virus as the major indicator of infection, and some tests to detect HCV antigen or RNA of virus as confirmatory tests. So for treatment, again, we have several drugs. It is sofosbuvir, and usually it is combined with letetresfovir or daclatasphere. And nowadays we have some combined drug. Previously they were available only as a separate components. People was needed to buy two drugs. And nowadays they're combined together, and that picture few examples of combinations are provided. And usually the regimen again is recommended by WHO, and course of therapy is approximately 12 weeks. So we talk already that treatment is more expensive, and till current day treatment of hepatitis C infection, one course costs more than $100,000. Usually can reach $150,000, but again, that course can eradicate infection completely and further treatment is not required. And in the contrast with hepatitis B, when lifelong therapy can be indicated, here it is a single course. So still, it is very significant number, and in some countries, in Russia included, we have some programs when government afford treatment and they can pay for the complete course, but list of requirements to get into that program is very difficult. It should be absolutely social man without previous negative histories with the law. It should be responsible person with job and stable position on the job, and huge list of requirements for those people, but it is possible to get into the list and receive the treatment paid by government. But of course, usually people don't have those money, and infection can become chronic without proper therapy. So for hepatitis C, we don't have any vaccines. Few candidates are registered for development stage, and some studies are going on, but no data on use on humans is available till the moment. And we have again only non-specific measures of prevention. They are common for hepatitis B and hepatitis C, and any hepatitis which is transmitted through blood. It is common measures of doctor's protection, use of gloves, don't perform any examination, don't touch any liquids of subject, samples of subject without gloves. Blood should be disposed in proper manner, and of course, sterilization of medical instruments and other things applied for that thing. Of course, barrier contraception during sexual contacts, even if chances less than in hepatitis B, but still common prevention is applied for any case for any infection. And the last virus of hepatitis, we will discuss hepatitis D virus, or known as well as Delta hepatitis. That organism actually is not really a virus. It's not classified completely till nowadays. It is awaiting taxonomic classification because actually we have viroids. It is a small pathogens there of subviral level, and usually they are identified in plants. So that human virus, Delta virus, is similar to viroids, but structure is more complex. But structure of virus is still not so complex as in viruses, and kind of an intermediate position. It's not clear is it viroid or virus. And a lot of discussions, a lot of studies are going on, but for several years, virus is not classified in corresponding manner. So virus of hepatitis D is the smallest pathogen of human, and it has the only antigen or the only protein which is coded by RNA of virus. It is HDAG, hepatitis D antigen. And actually, Delta virus required hepatitis B virus as a partner for infection. It can cause two types of infection. It is co-infection with hepatitis B when two viruses are transmitted from the same person at the same time. Or it is superinfection when one subject is already infected with hepatitis B and Delta virus will add to that infection. In both cases, virus of hepatitis B needs HBs antigen of virus hepatitis B for transmission and for entering the cell. It cannot invade the host cell without hepatitis B virus. So usually symptoms of Delta hepatitis appear as worsening of already existing hepatitis, or the main symptoms of hepatitis B infection will prevail, and virus itself is not distinguished between other diseases. So it just can join the infection, and all symptoms of previous or existing hepatitis B will prevail in the clinical presentation. So according to official data, about 15 million people worldwide are infected with hepatitis B and hepatitis D viruses. So again, we don't have any treatment and any specific preventive measures for hepatitis D, but if you will prevent infection with hepatitis B, hepatitis delta cannot cause any infection. So hepatitis B vaccine to prevent hepatitis B infection is the way to prevent delta hepatitis as well. And of course, eradication of hepatitis B infection, if it is possible, if it is acute hepatitis, will prevent infection of hepatitis D or cure infection with hepatitis D. Structure of viruses are quite primitive. Again, we have 3 genotypes and different distribution and worldwide. But structure of virus is identical. It is linear one, a line of RNA in the center, packed into the capsid and surrounded by envelope of hepatitis B. So the same small, large, and medium proteins are identified, the same coverage, and actually HBs antigen can be identified on the surface as well. So common questions about immunobiological preparations, they are identical not only for hepatitis E, here which I showed you hepatitis B surface antigen kit for screening of people, and it is ELISA kit targeted to identify HBs antigen. We have sets in the right picture. It is antibodies to hepatitis C virus. We have similar tests for HB, hepatitis B virus as well. We have ELISA test, ELISA kit to identify immunoglobulins against hepatitis D virus antigens. And actually, if you get that question, you just need to think what is present in that microtiter well, and what do you need to find in your subject. So actually, if it is key to identify surface antigen of hepatitis B, for example, we need to say that plastic wells are covered with antibodies against HBs antigen, and describe which options are possible. The subject is healthy, subject is ill, what will happen, how to relate the result. The same manner here, you can have kit to identify antibodies against delta virus, delta hepatitis virus. So just think what to take from the subject, what to look for, and what should be in those wells. In any case, system working by formation of complexes between antibody and antigen or anti-antibody, and the result indicates changing of color indicates positive result, and if color is not changed, then negative. Just describe the principle of ELISA kit, ELISA test, and apply it to current question. So that's all about viral hepatitis. I will not discuss deeply because actually it's not the task of our department, but common information about clinical course, structure, and diagnosis of virus is sufficient for you to pass the exam.